Welcome back to Biocraft. Today we will talk about the final step of gene expression and protein synthesis, translation. Just like in our last episode, we will explain this process in a series of steps that enzymes, code and amino acids have to go through to correctly synthesize a protein. Now, let's begin. Go ahead, Cedillo.
Before getting started, let us introduce the most important characters of RNA translation. First of all, we have the ribosome. We'd rather call it protein factory because that's pretty much what it is. A ribosome is in charge of hosting the tRNAs and the mRNAs in order to assemble chains of amino acids. Second, we have the transfer RNA, tRNA, which connects mRNA codons to the amino acids. In case you're wondering, codons are a specific group of nucleotides in each code for specific amino acids. There are 61 codons that are specified for one of each of the amino acids. Finally, we have messenger RNA, mRNA, which is a transcribed and processed information from a gene. It's single-stranded and has uracil instead of thymine.
Now, let's talk about the initiation complex. The process begins with tRNA binding to the ribosome. Let's remember that the ribosome is made up of two parts. These are called the large and small subunits. Then the ribosome and tRNA assemble onto the mRNA to begin the first scan. Since mRNA is not ready to be decoded, one of the tRNA carrying a methionine amino acid reads the first codon in order to initiate the synthesis of the chain. That pink circle represents methionine, and the red shape is a tRNA.
Second, we have elongation. As its name implies, it refers to the chain of amino acids getting longer. As you can see in the model, we have a growing chain above the ribosome. In this step, the ribosome and the attached tRNAs walk along the mRNA in the 3' direction, reading each codon individually and assembling the specific matching amino acid.
As you might see, the last step is called termination, which happens when the finished polypeptide is released. It starts when a stop codon, which could be either UAG, UAA, or UGA, enters and is identified by a tRNA. Eventually, the chain is allowed to exit the ribosome after a series of chemical reactions.
Like every good book, translation has also an epilogue. Although we have our polypeptide, this doesn't necessarily mean that the work is done. Our polypeptide has to join other polypeptides in order to form a 3D protein structure.
Thanks a lot for watching the video. We hope you enjoyed and learned a lot about DNA and how it works. See you till next time.